আপুনি জইন কৰিলে নেকি বাৰু? Hello, Dr. Das.
মই জইন হৈছো ৰ'ব। Am I audible?
Dr. Das.
Am I audible? Okay, fine. Can you start now?
ভাস্কৰজ্যোতি ভূঞা ন?
Well, should I start or wait? I assume that is yes. I mean, it's time for me to start.
Within a few seconds, we're going to start now.
Okay, sure. Please let me know.
Okay, we'll start now after a few seconds.
Sir, please come onto the dais and we request you to kindly take your seat.
Recording sir.
We would also like to request the coordinator of the session, Dr. Dilip Kumar Dekar, sir, President, Gurgaon College Science Forum, to come onto the dais. Now we shall proceed to felicitate the chairperson. I request Dr. Dilip Kumar Dekar, sir, to kindly do the honors. I also request our faculty members to kindly assist in the felicitation ceremony.
ভিডিওটা আরম্ভ হবে আরম্ভ হবে
Thank you, sir. Thank you, ma'am. Let me now introduce the chairperson to the audience. Dr. Bhaskar Jyoti Sharma, sir, is an assistant professor in the Department of Chemistry at Jorhat Institute of Science and Technology. Dr. Sharma is a highly qualified academician with an impressive educational background. He completed his Bachelor of Science from Juniper University and then pursued his master's degree from Guwahati University. He completed his PhD from Juniper University in 2009. Along with carrying out multiple administrative assignments, he has also successfully completed several projects under him. With his strong academics and expertise, Dr. Sharma has made significant contributions in the field of science. His extensive research work has been published in several reputed international journals, and he has also presented his research findings at numerous national and international conferences. We are indeed privileged to have Dr. Bhaskar Jyoti Sharma here today. Now, if the chairperson and the coordinator would kindly allow for me to introduce the first speaker of our invited guests, I shall proceed to do the same.
ভিডিওটা কি ইউটিউবের ভিডিওটা
Thank you. So we will start our first technical session. Organized by the forum people over IPC. Program college. Yeah, Senior Manager, Process Chemistry, Center of Excellence in Chemistry, Applied Materials, California, USA. Thank you.
Thank you. Thank you for.
Can I give a brief introduction about it? Dr. Bhaskar Jyoti Bhuiya, sir, is joining us from San Jose, California, USA, where he is presently a senior manager in process chemistry, applied materials SPLA, USA. He did his master's from IIT Guwahati and PhD in chemistry from IISc Bangalore. Other than being the organizing committee member of ECS Symposium ALD section, he is a reviewer of several esteemed journals published by World Society of Chemistry, Delhi, American Chemical Society, etc. Dr. Bhaskar Jyoti Bhuiya, sir, has in-depth knowledge and expertise in key semiconductor integration processes such as atomic layer deposition, area selective deposition, among others. He has also worked to solve challenging integration and process development schemes in key inflection areas of NAND, NAND, DRAM, MEOL, FEOL integration theory, along with 600 plus citations for publications in peer-reviewed journals, book chapters. He also has 20 plus granted US patents and 40 plus published US patents. With this brief introduction, I hand over the platform to you, sir, for your talk. Over to you, sir.
Sure. Thank you so much for your kind interaction. Really appreciate it. And good morning, everybody, and good evening, good afternoon. I mean, wherever we are located in this globe, right? Today, I'm going to talk about. I mean, you can see the role of chemistry in device miniaturization. That's mostly like about. It's related to the semiconductor manufacturing. So this I put. Am I audible?
Video.
All good, right? My audio is fine, I guess.
Yes, sir, you are perfectly audible. You can continue.
Sure, thank you.
Okay.
Yeah, so I mean, by training, I mean, I am a chemist. Actually, I'm a I did my PhD in like bioinorganic chemistry, and by luck or whatever, I moved on to the semiconductor industry back in 2010, right? And then, so I mean, today, I mean, I'm not going to talk a lot about the actual integration. I mean, I'm going to talk a little bit about the integration, but mainly the focus would be how, I mean, why is the chemistry so important, right, in this field? So yeah, I mean, I'm not an expert in device physics or the engineering, but at least now after working in the industry for around 10, 11 years, I have some feel about, or at least some basic understanding. So I'll try to put the talk in such a way that like a very generic talk, right? I mean, if there's any question or any specific thing that anybody wants to know, you can reach out to me. I mean, ask me, stop me. I mean, during the talk, or maybe ask me questions after the talk, or maybe like you can shoot me an email later on. So yeah, having said that, right? As I said, I mean, we are like, I mean, we have participants from US now. We have a few of my friends are from also signing from Europe, right? I mean, this is, I mean, back in 2010, I think it was not so such a common feature, right? Usually, I mean, I think we had Skype back then, and like, and then like we never thought of having a meeting like this. I mean, in a global with a global presence. So all this is possible. Definitely, the software companies are working amazing, like Zoom. I mean, Teams. We have all those meeting platforms, but without the hardware, I don't think this would have been possible, right? And this is where the semiconductor manufacturing comes into play. So this is a very generic slide. I think there are a lot of experts in these fields. I mean, semiconductor. Just a minute, something is disturbing my screen. Yeah. So it's basically, I have just put a picture of a transistor here, right? When I grew up, like in my high school, in my physics classes, like we used to have the 3-pin thing, and the 2-pin is the diode, right? And then 3-pin is a transistor. But this is a very, very essential. This plays a, I mean, over time it evolved, right? I mean, it was like a 3D, like in our old radios and TVs, we have those transistors diode that you can see, right? Now it comes in a chips. So this is all possible because of the semiconductors, right? So in semiconductor in general is like most of you know, like it's intrinsic and extrinsic, right? I mean, intrinsics are like silicon, germanium, and then extrinsic are like either you dope with it like p type, that is like electron rich, sorry, positive type, so it's like electron deficient, like elements like boron and like group 3 elements, right? And then if you dope it with electron rich element like phosphorus, arsenic, so it becomes n-type, right? And then over time, as I said, it has evolved, right? And then from this kind of structure, I mean, this still exists, but still, what I'm saying is when we grew up, as I said, this is what a transistor looked like, and now it has moved to like thin films. It's the same concept basically. You have a source, right, and a drain, and there is a gate oxide or gate. It basically, when you give an electrical signal, this gate allows you to flow electron from so these are all positive, but my mistake. So basically the electron allows it to flow current, right? And that's how you like flow the signals in the circuit. And as a result, and this is basically the core of the ICs, the integrated circuits that we see all around. And this has not happened overnight, right? So, Gordon Moore, he was the co-founder of Intel. In 1965, he predicted that the number of integrated circuits, we call it a law, but still this is just in kind of an observation or a remark kind of, right? So it's basically every year, the number of transistors will double in a integrated circuit. And that is what he predicted in 1965. And if this is just a plot of the number of transistors those are present, and this is the x-axis is your time, right? This is up to 2020, 2018 time frame. And this actually rule more or less follows, right? I mean, there is a linear dependency, like every year kind of it doubles. So what does it mean, right? In terms of the normal lifestyle, or like what we say is in terms of how these devices, semiconductor devices are made. So basically it has, as a result, of this, your real estate, or basically the area given at a given area, you are increasing the number of transistors, like doubling every year. So it's basically an exponential growth. And as a result, you have to, like, scientists work like a group, it's like not one very, it's a huge group of people, right? Starting from, like, say, design engineers to material engineers to electrical, mechanical. You can just think of like almost hundreds of thousands of people working in this domain over like whatever last 30, 40 years, right? And with their hard work, you can see that this is the minimum size of a device. Like if you go back to the previous slide, as I said, the transistor has like the N type and P type, right? And there's a gate. So that, say, the critical dimension of the nanometer node that we talk about, this is the critical dimension of a transistor. And so this has shrunk over years. In 1997, say, I'm not going back to earlier node. This is again by the way, just a disclaimer, like all the information that I have is, most of this you can get it in Google, right? So, or like you can get it in Wikipedia or like some very open sources. So there is no confidential information, or at least by intention, I did not put any confidential information from anyone. So just to let you know. Yeah, so this is the dimension. Like in 1997, I tell you what, this started with 250 nanometer. And just for reference, right? This is the scale that nanometer looks like. And an atom is like 1 angstrom level, right? Around an angstrom. So, and molecule, it typically is like few nanometers, maybe a couple of nanometers. And then say DNA, the width of a DNA is like what, around 10 nanometer? If I'm not so wrong, or 7, 8 maybe, right? This is basically the scale. And I think the human hair falls somewhere in the micron range. So micron to nanometer, this itself was a huge achievement at that point of time, having a critical dimension of 250 nanometer in a IC, right? And then, or in a chip. Then it kind of, it's again, don't go by exact number, but it's not like exactly doubling, but yeah, it doubled because the dimensions shrink, right? From 250, 180, like it goes down. And by 2018, so most of the advanced chips are produced in this 7 nanometer node. And this, why is how is it possible, right? As I said, initially everything was controlled by engineering control. Like you have a very precise engineering control to make the dimensions shorter, like in some nanometer nodes, like, and then eventually it was not possible, right? So if you look at the periodic table here, so this gives you the importance of chemistry in this industry. Gives you an idea, right? In 1980s, there were like handful of elements, like around maybe 15, 20 elements in green. Those were used in this industry for wafer manufacturing. And then by say 2000, this was the actually, there was a huge shift in like new chemistry incorporation in this field. And by around 2000, almost like half of the periodic table is used, right? And by now, I would say around 70 of the elements are used in wafer manufacturing at different stages. So that's why this is the backbone of the whole talk, right? Chemistry plays a vital role in semiconductor manufacturing. And so the expertise in chemistry, I mean, definitely engineering is important and is the core, but for the process side, chemistry is also very, very important for this industry. So again, this is just a representation of what we have seen, at least I have seen, or most of my colleagues and friends. I mean, almost everybody can relate to this, right? As I said, in your high school, we saw the transistor like big 3 pins coming out. Again, this is the transistor. There are diodes, there are different logic circuits, and I'm not going into all the details. And then back then, like we had use for these floppy drives. I think most of us are familiar with this, right? And that time, I think it came out what, like few kilobytes of memory, maybe hardly like an MB, or it was like very, very small compared to the size of the memory devices that we have. So which, I mean, this small flash drive can hold like terabytes of data, right? And back then we are talking about like 20 or 25 years back, it was just this big floppy drive had only like few kilobytes. And this is again re-emphasizing the fact, right? Basically, that how there is a tremendous innovation that has happened over time. And how is it commercially feasible, right? That is another thing. I mean, you can have these are like very highly precision engineering that tools that we need to use. So the cost of the tools are like really, really expensive. But I mean, if you look at the now, I mean, this is our data. I mean, the cost of the transistor basically cost of transistor per transistor in a chip, right? So when the number of transistors increases, the cost per transistor actually decreases. So when I joined the industry, that back then we were saying like when iPhone was invented, right? iPhone 3, 4, the amount of transistors that it was there in those iPhones. If we would have to use the transistors that were available back in 1980s, it would have been like a 2-story house or something like that, right? So because of this miniaturization, like in one given area, we are increasing the number of transistors. So basically, this is possible. So again, this is just like another generic thing, right? We all know the shift in the phone industry, right? So we had some like these big phones, then this pocket phones like flip-ons, right? And then we have iPhones and the smartphones that came up. So basically, you are increasing the performance of the chips, right? So we also need like very high power in terms of battery or whatever, like the computing power. And and then the dimensions are shrinking and the cost per transistor is shrinking. So this is the scaling that has been done in over last 20, 30 years. And this is going on. And this is, as I said, like possible only because of the miniaturization. And this is, in this slide, I want to show you the core of a chip, right? Again, there are different aspects of looking into it, right? So this is one of the published thing. So it's basically at this level, you have the core transistor that is there. This is the silicon area here, and then you have the gate, right? After the gate, you have the first metal contact, right? And then we have, this is where the whole, basically this is just an expansion here in the right top. So this is the main contact, and then you have the lower end interconnects, and then you have the upper end interconnects, right? So by definition, like we call it like front end is where this before the metallization, then the middle end and the back end integration happens. So in other words, if you look at the integrated circuit, so it is basically, you can say I mean, transistor is basically, as I said, like it processes a signal depending on how you, I mean, give the initial voltage to either it's like zeros and one, right? So basically you have electron flowing from 0 to 1, so you pass the signal. And then in today's world, we need a chip or for the current processing power that we need, right? So it has to be able to do like multiple functions at the same time. So as a result, you can think of this as a like a transistor here, and there are like millions of flyovers over Like where if say a signal has to go from A to B, if it cannot take part one, so it will go to the other one or else the other one. So basically it's a network of electron pathways that can be pathways, I mean, or highways where the electrons can flow, right? And that's where whole backbone of these chips are, right? It's the metallization part that is, I mean, like there are other critical, very, very critical parts, but metallization is one of the key component in a chip. And where are we in the real world today, right? So this is like data published from Apple, right? So iPhone 12 or higher version 13 like uses A14 chip. And look at these astonishing numbers, right? So you have, as I say, the transistor is somewhere here, and there is a billions of pathways here. And in this chip, which is like 88 square millimeter, it's 8.57 mm by 10 mm, right? So we have 11.8 billion transistors, right? This is what the innovation means, right? I mean, everything in that, and this is the technology like where we are talking about beyond 7 nanometer, right? So you're scaling beyond. So like 7 nanometer is when like 2018 was in production, and this is in 2022. So you can just imagine. So we are coming to a something like an atomic limit, right? For this, like making the chips. And how does it work, right? Now getting to the main thing. So we are lucky that silicon is the starting point, and silicon is the second most abundant element in the earth, right? So from this silicon, we make a silicon, like we get a pure silicon, then make a wafer, and then they make the chip. And the whole talk or most of the processes that I'm going to follow on from here is what happens in the wafer, right? So it's basically, it undergoes a lot of steps, like starting from how it is done, then oxidation, then initial patterning. I have some subsequent slides where it is slightly explained more. And after this, actually after the patterning, there are a lot of deposition steps that happen, deposition, etching, CMP, and all those. So this is where, I mean, this is a huge industry. I mean, as I said, so there is a lot of opportunities here. And in 2021 itself, we call it the WFE, Wafer Fab Equipment Manufacturing, which our company does. We make equipments to make the chips. So we don't make any chips by ourselves. So those are mainly made by our customers like Intel, Samsung, TSMC. But we make those machines where we deposit say a film like so precise in the atomic level that it needs like new chemical innovations. I mean, innovations in the field of mechanical, electrical engineering, everything, right? It's basically a combination of and hard work of a huge team of people. And most of these, not most, almost all this work is done in the clean room. So where, because the contamination, because we are talking about like 5 atoms sitting on top of each other in a very orderly manner, right? So if there is any particle or anything that comes in, so it's a huge problem. So all work are mostly done in this clean room, and you can just see an image of a clean room, right? So people are all gowned up. So even your um, like particles from your body hair, your sweat, everything can create a lot of problem in like defects. So a very highly clean environment is maintained in these rooms, and then all the manufacturing processes goes on. Again, this is another slide, like what happens to silicon. So this talk is more about chemistry, so maybe people might be slightly interested, right? So the way it works is like we melt the silicon, right? And then they make something called ingot. So these are like different diameters depending on what we want to make, and then they slice it, they make the wafer, it goes to the tool, and this is what comes out, right, as end product. And so depending on the quality of the processes. And by the way, to get to this step from here, say, it a modern chip, a chip that is used in all this current era, or undergoes like around 400 plus precise steps, I mean control steps, like starting from patterning to deposition, and then finally it gets like diced. Basically, you cleave these dies, and everything, like every square thing, repeating thing that you can see here is a chip. So they after that they're cleaved into like particular die or chips, and then it goes for packaging where you can see those bumps at the backside, right? So you do the like soldering and basically make the uh, uh, basically that's what we see as the end product, right? As an end user. So as I said, it undergoes a lot of steps. So this is just the basic thing. So on silicon, it's a planar silicon. It's very well polished. Then we do a, say, this is patterning step. So our company doesn't do a lot of patterning, but still we do some. So it basically undergoes a hard mask, then they do spin coating, then the initial pattern is formed, right? Then they do an edge, then they strip off the hard mask, right? You end up getting the pattern that you want, and finally basically it undergoes a series of steps, right? And finally we end up getting a step like this becomes a like core, and then you do say there are a lot of deposition, etching and polishing steps that undergoes after that. I mean, as I said, only in this space I would say the wafer undergoes at least like 200 to 300 steps. And then going into slightly more into chemistry, right? So thin film deposition technique, as I said, I mean, it can be divided into like broadly 4 categories. Sorry, physical vapor deposition, which is more of like you have a very pure metal or an element in a machine, and then you can you can excite that with like high energy, so it evaporates and then it deposits on the, like basically if you physically make it vapor, right? With like so these are some of the techniques like thermal evaporation, pulse laser evaporation, and all those, and sputtering. So it's basically non-selective, or whenever it forms and it comes and sits on the wafer. So up to certain age, for certain application, these are like awesome. But when you are, yeah, was I heard something? Okay, so it works, but then like it's mostly like engineered control, right? And then for the next era, it came the chemical vapor deposition, which is more of like you need like organometallic precursor or organohalide precursor, right? So so that's where the chemistry comes into play more. And then there is another technical atomic layer deposition, which is mostly like chemistry dependent. I have another slide on atomic layer deposition. We can go slightly more into the details. So these can be like again thermal ALD or plasma enhanced ALD. So if you talk about ALD, take it this way. I mean, this is like we are talking about depositing, say, 5 angstrom or 10 angstrom of a layer of a material, say aluminum oxide, precisely over that 300 wafer that we usually use for these processings, right? So you can imagine that, like in the map of India, we are trying to deposit, like say the 10 angstrom or 20, 50 angstrom precise layer within uniformity, by which I mean like if it will go out of the boundary of the Indian map by say a few millimeters, that is the precision level that we are talking about in this kind of deposition techniques. And obviously there are other techniques, the solution-based techniques like Sol-Gel. A lot of you might be already aware of this, right? Spin coating and screen printing, etc. So this is a slide. It's mostly on the atomic layer deposition. So this is the perfect example. So this tech, I mean, there was a lot of innovation done after that. But the cool thing is that this process is self-limiting. So that means if you have like excess of, so this is called TMA, trimethyl aluminum, and then water, right? So we pulse in a huge pulse of TMA to the surface. Only it forms only one layer at the silicon wafer or the wafer or the substrate, right? Then we purge all the unreacted species out, right? And then it basically forms a monolayer of aluminum, and then water comes in. You do a huge flux, and then whatever has to react with TMA, this is like a I mean, very highly pyrophoric chemistry, right? So it will definitely react with water to take the aluminum carbon bond becomes basically ALO, right? And then you deposit one layer of aluminum oxide after the purge. So this is very helpful for depositing a very uniform film around the topographic structure, because in the initial slide I mentioned, right? So it becomes like you have like you do a patterning on the silicon itself. So this is like one of the best technique to do a very conformal film. But obviously, limitation for this is like it has a very low throughput, because I mean obviously there is some like throughput is one of the key thing that we look into, our industry look into, like whenever we are designing a tool or doing a process. So yeah, there are ways of improving it, but yeah, there is some limitation with throughput is one of the primary limitation for the ALD process. But think about this, right? I mean, this process has led to another innovation in this industry. So this is a very hot topic in the semiconductor field right now, processing field. So selective deposition. So imagine like these are like, again, the dimensions, these are not scale, but I'm showing these dimensions somewhere around like say few nanometer, like tens of nanometers maybe, right? So selective deposition, I mean, this is like an example of metal on metal. This yellow is a metal part, and this red is a new incoming metal. So you can, is like there are ways of depositing a metal, like say the width of this channel is around 10 nanometers. So you can precise a deposit another metal on top of that at that 10 nanometer width level, right? So this is like, I mean, that's something that we cannot see in our with our eyes, right? We have to image that with TEM and SEM and whatnot. Um, but this is amazing, right? So there is another application for the selective depth we do have, and these are some technologies that we worked on and we have developed over time. I mean, we as a team, like as a team, as a company, as like whole industry in general, right? So you can also, there are ways of doing a selective bottom-up fill. Like if you're doing a PVD or a deposition here, so theoretically it should deposit everywhere, right? So basically, like on this blue part as well as in this yellow part. But there are ways of controlling this, like through processing and new chemistries, that where we see a bottom-up growth of the metal, or like or dielectric, whatever we're using. So there is another one application like the selective sidewall deposition, like you can deposit. So this is say a metal, this is some dielectric, and you are depositing a sidewall only on top. I mean, why we need it? I mean, these are like slightly, I mean, we can talk more in details later if anybody is interested. But I mean, there are ways of doing this kind of deposition as well. And this is all possible mostly because of the innovations in the field of atomic layer deposition. So one of the best thing about the selective deposition, I mean, that enables is the self-aligned via growth. So this is something like, say, think about this, right? I mean, we have the pattern, we have the dielectric here, we have the metal here. So and in the previous slide I have shown, right, on the metal, you have to grow a metal again. So for that, you have to do a lot of patterning steps. So selective deposition can enable, and then when you do those patterning, so these are like critical feature right now is like around 5-6 nanometer, 10 nanometer at that level. So you have to do a masking. If your mask doesn't overlay well on top of the layer that you want to mask, right? So you will end up getting like a mask misalignment. So if I'm saying that you want to have this mask here on top, so it will, if it go, and this dimension is say 10 nanometer, right? So if it is displaced by 2 nanometer this side, you are essentially talking about an area of 8 nanometer here, right? Where the contact will come in. So that means you're already reducing your contact by 80%. By contact, I mean, it can be a dielectric to dielectric contact as well. So that's what I mean here. But you can just imagine, right? So, but if you have a selective way of depositing that selective layer, then you don't have to limit, I mean, worry about that, the mask misalignment issues, right? So this is another huge inflection like people are working on now. And in terms of selective deposition, I mean, there are like, this is just one slider on like, it can be, there are two approaches. There can be something like inherently selective, like most of it is say metal on metal, right? Metal has an affinity to another metal. So using the process control, using the right chemistry, the precursors, we can, there are ways, and we are the pioneer in doing this selective metallization approaches, right? So you can grow say selective cobalt on copper and things like that. So there is another way of doing selective deposition where you can induce the selectivity. So this is mostly done with self-assembled monolayers, or now the buzzword is SMI, the self single molecule, sorry, small molecule inhibitor. So assumed as this, right? I mean, this orange is a metal, this gray is dielectric. So by selective treatment, so everything is like dielectric mostly is like silicon oxide based, right? So silicon oxide has an affinity to a certain kind of say group. So this chemistry goes and binds only to silicon oxide, right? And then when you deposit the next layer, this green layer, say another dielectric or metal, whatever, another metal. So you basically end up depositing the metal on top of the metal. So this is like we call it the induced selective depth. So it can be used to do metal on metal or dielectric on dielectric. Oh, sorry. So yeah, I'm almost to the end of my talk. So this is what I mean. This industry has been like always going in a cyclical mode, at least in like 10 years back, right? But lately, yeah, I mean, the future for this industry is bright. The sense like initially it was always dependent on some inflections, like say PC and internet era, in like back in like late early 2000s, right? So PCs and internet these were booming. So there was a spike in the growth, like demand for this industry, and then it again fell. Then the mobile industry came in, right? So I mean, you have say from iPhone 3 or 4 to like right now it is what iPhone 14 or 15 is coming up, I guess, right? And then, uh, but the next inflection for the semiconductor manufacturing industry or WFE or basically semiconductor industry in general is that all the interest that we are like getting for AI, big data, right? So you need like for AI, you need like very fast processing processors. You need like a lot of memory to store all the data that we are collecting, all the artificial. I mean, for all these inflections, right? So I think there are. This is just I mean, this is the in general industry thought, right? I mean, there would be a huge demand for semiconductor devices in next 5, 6, 10. I don't know, like it's going to go up hopefully, and that's what we hope for, right? And this is just some related numbers here, right? In by WFE, I mean wafer fab equipment manufacturing equipment. So the market size for this one is like around $70 billion in 2021, and which is going to go up to like another 100 billion in next whatever 7, 8 years, right? With a annual cumulative growth of around 8%, right? So I take yeah, and then this is just like a slide, a glimpse of what we do as a company, right? So I mean, we have systems like where we can do a lot of processes without any vacuum break. I mean, this is because vacuum is the killer. Vacuum break is a killer in a lot of processes, right? So this is just an example of one of our platforms. So when you can do like different flows like ALD, PVD, CVD, I mean, like different processes that can be transferred from one chamber to another. These are like 300 wafers or 12-inch wafers. So like we can do different processes here, right? So yeah, that's it. I mean, I'm almost at the end of my talk. I don't want to read through it. This is just like, as I said, this is a industry where a lot of innovations has been made in last 30, 40 years, and a lot of things to come up, right? And demand for semiconductor is not going to go down because of the huge inflections those are coming up, and AI, big data, and like even the EVs, right? I mean, recently I read an article like somebody was saying that there's like 400 plus chips in a Tesla now, right? Or maybe more, or 4,000, forgot that number. But I mean, so those are may not be the high end in all the cases, but say your rear view sensor needs something. Your like there are a lot of sensors everywhere, right? So I mean, you need a chip to control all those. So because of all these demands, so this need for this industry is going to go up. And I don't know. I mean, like if somebody is interested, right? So like you can reach out to me. I mean, we can talk more. I mean, what are the career options that we have in this industry? But definitely, chemistry is a key. Chemistry plays a key role. Has played a key role uh, in the innovation in this field. I mean, I'll just like to conclude by saying that if anybody has any question or anything, feel free to ask. Or if we're running out of time, we can talk later. That's it. Thank you, everybody.
you.
Thanks for your.
Thank you, Dr. Bhaskar, for your nice presentation. And now the session is open for discussion. If anybody has any question, you may ask. I think no one has any query, but I want to say something. Bhaskar, can you hear me? Am I audible?
Yep.
So do you remember I met you in IISc Bangalore in 2007, I think. You were there in 2007 in IISc?
Yeah. I was in IISc from 2005 to 10.
Yeah.
Yeah, I did not get
IISc in connection with some symposia.
Okay, got it. Actually, I did not get who is talking, so I did not recognize your voice. Bargaon College.
I'm giving it to you and thank you for your nice presentation. Thank you very much.
Thank you. Thanks for having me.
Okay. So now the second speaker of this technical session, Meenakshi Saikia. I request Sahil to introduce our second next speaker of this session, Dr. Meenakshi Saikia.
Our next speaker, Dr. Meenakshi Saikia ma'am, is a postdoctoral research associate at Radiation Oncology Department, Washington University School of Medicine at St. Louis, Missouri, USA. She did her PhD from Rajiv Gandhi Center for Biotechnology, and her current research focus is on understanding the signaling events that orchestrate the process of mid-gain upregulation upon TIP1 blockade, a novel molecular target in NSCLC. She has several publications and review articles in reputed peer-reviewed journals and books. She is a gold medalist from Dibrugarh University and recipient of DST Inspire Fellowship, and she has also received Dr. M.R. Das Research Excellence Award in recognition of professional achievements during her PhD tenure. She was awarded the Best Poster Award at the INCD International Conference organized jointly by Translational Cancer Research and IIT Roorkee in Kerala. She also has participated as JSPS Hope Fellow at 7th Hope Meeting with Nobel laureates at Japan Society for Promotion of Science in 2015. We welcome Dr. Meenakshi Saikia ma'am and request her to deliver her talk.
Good morning, everyone. Hope I'm audible.
Good morning, ma'am.
Yeah. Okay. Hello, everybody. Thank you for the kind introduction. The title of my today's talk is Chemosensitization and Molecular Targeted Therapies for Cancer Treatment. During my talk, I will walk you through two sections. The first section will comprise of where I will talk about chemosensitization, where I'll talk about identification of triptandrin, a natural compound, as effective chemosensitizer for melanoma chemotherapy. This work was done under the guidance of Dr. Rubijan Ento during my PhD at Rajiv Gandhi Center for Biotechnology, Trivandrum, Kerala. And the second part of my talk will be on molecular targeted therapy, which is my current assignment, which I am doing under the mentorship of Dr. Vaishali Kapoor at Washington University in St. Louis School of Medicine. And there I will talk about the mechanisms of mid-kind upregulation on blocking the functional domain of TIP1 for lung cancer treatment. Though this topic sounds a little bit technical, I'll try to keep it as simple as I can. Well, coming to the first part, that is chemosensitization, let's begin it. So when we talk about cancer therapy, there are different strategies that are adopted, and out of which chemotherapy is the most sought out mode of therapy. But prolonged exposure of any chemotherapeutic drug results in chemoresistance due to upregulation of various survival signals. Let me start the indicator. Yeah, so I'll explain again. So when any chemotherapeutic drug is exposed to the cancer cells for a prolonged period, a mechanism called chemoresistance is developed, which is due to the upregulation of various survival signals. Many substances, or which can be either chemically synthesized or naturally available, can downregulate these survival signals and can act as chemosensitizer. This process is known as chemosensitization, wherein this chemosensitizer can sensitize the cancer cells towards the chemotherapeutic drugs to enhance its therapeutic efficacy. As a result of chemosensitization, the dose of the drug, the side effects, and the cost of the drug can be reduced. In our lab, one of my seniors had identified DWF5 from the leaves of the plant Rhytia tinctoria, which is a semi-purified active fraction. And Rhytia tinctoria is basically a herbal medicine whose leaves, bark, roots, and seeds are used to treat different malignancies such as psoriasis, dandruff, and other skin infections. So we used this DWF5 extract and checked its efficacy against malignant melanoma cells. And we could find that it can be used against malignant melanoma. This work was published in scientific reports. And then we further purified this semi-purified fraction. We could find that it comprises of an aliphatic fraction and an aromatic fraction. The aromatic fraction was identified as triptandrin. So my goal was to analyze or study whether triptandrin can be used as a chemosensitizer for dacarbazine chemotherapy, which is a common drug used for malignant melanoma. I tried several combinations of these two, that is dacarbazine and triptandrin, and I could find that particular concentration, that is 50 micromolar dacarbazine with 2 micromolar of triptandrin, could induce a synergistic cytotoxic effect in the melanoma cells. As you can see in this figure, we can see that the combination of triptandrin and dacarbazine is inducing cytotoxicity in the malignant melanoma cells more as compared to the drugs alone or the control. We used assays like MTT assay and thymidine incorporation assay to initially identify the combination. This is where we can compare the number of cells, that is the relative cell viability. Here you can see that the combination is reducing the viable cell population significantly when compared to the drug alone. Next, we went ahead to study the mode of cell death that was inducing by this combination, and we could find that triptandrin potentiates dacarbazine-induced apoptosis in A375 cells. That is because it could induce the cleavage of caspases and cleavage of PARP. And we could also see that nuclear condensation was enhanced in triptandrin and dacarbazine, which all indicates that apoptosis was taking place in the cells when the cells were treated with the combination of dacarbazine and triptandrin. Next, we checked the different molecules that are involved in cancer upregulations, such as phospho-P4244, PJNK, P38, AP1, NF-kappa-B, etc. And also melanoma-specific molecules like BRAF, BRN2, beta-catenin, etc. And we could find that the combination of dacarbazine and triptantrin, when given together, is able to downregulate these survival signals, which is induced by dacarbazine alone. Next, we went ahead to study the in vivo validation of the chemosensitizing efficacy of triptantrin towards dacarbazine. We first studied the pharmacological safety of the combination using histopathology and liver function tests, and we confirmed the pharmacological safety of dacarbazine and triptantrin combination. Next, we went ahead and designed the animal study to study the anti-tumor efficacy of triptantrin-mediated chemosensitization of dacarbazine. So this is the experimental design of the animal study, wherein we injected the melanoma cells to the mice. And right from the next day, we started the treatment of the different drugs, either in combination or in alone in different groups. The drug was given intradermally for 4 weeks on a twice weekly regimen, and animals were sacrificed at the end of the experiment, and the tumors were excised. So as you can see in this image, pretreatment with triptantrin significantly inhibited tumor development in the melanoma xenografts in dacarbazine chemotherapy. So we could find that when the animals were given with the combination, the size of the tumor was significantly reduced in the set of animals that received the combination. So also, the cells which we had injected to induce the tumor in the animals had Luke genes transfected into them, because of which when we give the luciferase substrate and do the in vivo imaging of the animals, we can study the metastasis. So we can see that in the animals that receive both triptantrin and dacarbazine together, metastasis was significantly inhibited in this set of animals as compared to the other groups. So from this first part of the presentation, we could make this conclusion that triptantrin, which is the aromatic component of DWF5, could act as a successful chemosensitizer in dacarbazine chemotherapy. The combination is pharmacologically safe and induces a synergistic cytotoxic effect. Triptantrin could downregulate the several survival signals that were upregulated by dacarbazine in melanoma cells, and it could strongly inhibit melanoma tumor development and metastasis. Well, this was my guide during my PhD, and these were my lab members. So next, I will jump on to the other part of my presentation, which is the current assignment that I'm working on. That is molecular targeted therapy for TIP1 in lung cancer treatment. So discovery of new druggable targets is very important for the therapeutic advancement of the disease. And here we are looking at TIP1 as a druggable target. So let's begin with lung cancer. So lung cancer comprises of two parts, that is small cell lung carcinoma, which accounts for 15% of total lung cancer cases, and non-small cell lung carcinoma, that is NSCLC, which accounts for 85% of the total lung cancer cases. And we are here dealing with the NSCLCs, that is non-small cell lung carcinoma. So as I already mentioned, we have identified TIP1 as a druggable target, and that is because TIP1 is also known as the TEX interacting protein 1 or glutaminase interacting protein 1. TIP1 is highly overexpressed in various cancers, and it plays a role in tumor cell adhesion, migration, angiogenesis, and pulmonary metastasis. In regular terms, TIP1 is needed for various functions such as normal brain function and all, but it also plays a role in tumorigenesis or cancer pathways. It is a radiation-inducible antigen, and it has a single PDG domain, which is the functional part of the protein. And it is a small molecule. And often it is seen that high TIP1 expression could be correlated with poor patient survival. to understand the significance of TIP1, we created CRISPR knockout of TIP1, and we could see that the CRISPR knockout of TIP1 significantly reduced the viable cell population in a time-dependent manner. A549 is a NSCLC cell line, that is non-small cell lung carcinoma cell line. Then, since there is no inhibitor against TIP1, an anti-PDZ antibody was developed. That is, anti-TIP1 antibody was developed in our lab. And we found that when the cells were treated with this anti-TIP1 antibody, the cell proliferation was significantly downregulated in 2 different cell lines studied, that is A549 and H460. Next, we went ahead and did a global proteomic study wherein we processed our samples and subjected them to mass spectrometry. This allows us to study the fold change of the various proteins present in the cell. And when we did the analysis, we could find that MITKINE, that is MDK, is showing almost a 10-fold increase in the TIP1 treated samples as compared to the samples treated with the isotype. So we could find that there is an upregulation of MITKINE, and this was confirmed using Western blot in 2 cell lines, that is H460 and A549. We can see that when TIP1 was treated in the cells, MITKINE was upregulated significantly. So this led us to the discovery of MITKINE. What is MITKINE? It leads to therapeutic resistance. So often we see that when we are giving a particular drug, it stops working after some time. Why? That is because of this kind of molecules which give resistance to the cells. So coming to MITKINE, it is also called as neurite growth promoting factor 2. It is basically a small secretory cytokine, which is generally present in the body during the embryonic stage, during the mid-gestation period. It is basically in abundance. That's why it's named as MITKINE. And it is a secretory cytokine, basically. And in normal adults, its presence is minimal or nil. But when there is inflammation or any other malignancy such as cancer, the level of MITKINE elevates. It plays roles in different processes such as cell proliferation, angiogenesis, that is blood vessel formation, and epithelial to mesenchymal transition. And all these processes together lead to cancer metastasis, that is migration of the cancer from one part to the other. That means MITKINE is having a pro-proliferative effect. So my aim was to unravel or investigate the mechanism of MITKINE upregulation upon blocking TIP1. How MITKINE is getting upregulated when we are blocking TIP1, that I had to study. So my present slides will basically explain this, my upcoming slides, that means. So initially I started with studying the expression pattern of MITKINE in 2 different cell lines, that is A549 and H460, by 2 different experiments, that is real-time PCR to see the mRNA level of MITKINE and ELISA to see the protein level of MITKINE. So we could find that when anti-TIP1 antibody was treated in a time-dependent manner, we could find that the mRNA level of MITKINE as well as the protein level of MITKINE was gradually increasing in both the cell lines. We did various literature research, and we could find and pinpoint to come down to one particular pathway which we felt that it might be playing a role in MITKINE upregulation, which is the Wnt beta-catenin pathway. So coming to summarize the Wnt beta-catenin pathway, we can say that there are two stages where we can like two conditions where in one condition Wnt is off and one condition Wnt is on. Wnt is a molecule, and basically this is the plasma membrane, and these are receptors which are present on the cell surface. So when the Wnt molecule is not present, that means the receptors are free. The destruction complex is there in the cytoplasm, and it harbors all the beta-catenin and results in ubiquitination, and which leads to ultimately its proteasomal degradation. Whereas if Wnt is present and binds to the receptors, the destruction complex is harbored at the plasma membrane, and as a result of which beta-catenin is free in the cytoplasm and does not undergo proteasomal degradation, but it translocates to the nucleus wherein it binds with the promoter region and then transcribes various genes which have pro-proliferative roles. That means it aids in cancer. So based on the different studies and other literature research, we could design or propose a set of events which could be happening upon blocking TIP1. So the antibody, this is the antibody which we developed, that is anti-TIP1 antibody here in blue. And we propose that the TIP1 interacts with beta-catenin. And when we give the anti-TIP1 antibody, it takes up or blocks the TIP1. And as a result of that, beta-catenin is free in the cytoplasm. And then it can translocate to the nucleus, bind to the TCF-LEF site, and then transcribe the responsive genes, one of which is MYTKINE, followed by its exocytosis into the supernatant, or in human system, the blood. So first of all, we went ahead to check if TIP1 interacts with beta-catenin. We did different experiments, such as the co-immunoprecipitation experiment, wherein we could find that TIP1 was able to pull down beta-catenin both from the cell membrane lysate as well as in the cytoplasmic lysate. TIP1 was able to pull down beta-catenin. So we could say that TIP1 is interacting with beta-catenin. Another experiment we performed to check the same phenomenon was immunofluorescence using confocal microscopy, wherein what we do here is like, we have two molecules, right? The TIP1 and beta-catenin. We stain them with antibodies conjugated to different fluorochromes. Here, TIP1 is stained with red fluorochrome, and beta-catenin is stained with green fluorochrome. And this basically stains the nucleus of the cells. So when we merge the images, that is, we can see that there's co-localization of TIP1 and beta-catenin, which is seen by the yellow or the orange color. This is a 40x magnification, and this is in 63x oil immersion magnification, where we could see that the yellow is indicating co-localization of TIP1 and beta-catenin. That means these two are interacting at the cell surface. Next, we went ahead to see if we block TIP1. That means if we remove this triangle things, that is the TIP1 protein, is it inducing upregulation of beta-catenin transcriptional activity? So for doing that, we performed the TOF-FOP reporter assay, which is a classical assay to study the beta-catenin activation. So I'll briefly explain it. Here, we use a molecular biology technique called transfection. So we have two plasmids, that is TOP and FOP. These are basically same constructs, with the only difference is that the TCF-LEF promoter site is functional in TOP and it is mutated in FOP. So FOP is used as a control for TOP. So what we do here is like beta-catenin binds to TCF-LEF site. So these plasmids are transfected into the cell, and when beta-catenin is present in abundance, we will have lot of luciferase enzyme. Upstream of TCF-LEF site is a luciferase gene. So when beta-catenin binds and transcribes, we get a lot of luciferase enzyme being produced here. But here the TCF-LEF site is mutated. So even if enhanced beta-catenin is present, it cannot transcribe this luciferase gene. So as a result, when we give the luciferin substrate to this for this luciferase enzyme, we will get an enhanced signal from the cells that were transfected with TOP rather than the cells that were transfected with FOP, because there is less or no luciferase in the cell. So this is basically the TOP-FOP reporter assay, which is used to study the activation of beta-catenin, nuclear activation. And so this is the experiment which we performed. Basically, we did this transfection and seeded the cells in a 96-well plate, and then treated the cells with anti-TIP1 antibody. And then we did the bioluminescence assay, that is provided the substrate, luciferin, and did the assay at different time points like 0, 6, 24, and 48. And we could find that upon blocking TIP1, that is when we give the anti-TIP1 antibody, beta-catenin is activated at 24 hours. Next, we went ahead to check if MITKIND induction following TIP1 blockade is mediated by beta-catenin. So for studying this, we knocked down beta-catenin. We knocked down beta-catenin using 2 shRNAs, and this is the real-time PCR data showing the transfection efficiency of this shRNAs in A549 cells. And so here, basically, we want to see if beta-catenin is knocked down, can MITKIND be upregulated upon anti-TIP1 antibody treatment? So what we did is we did 2 experiments to study this phenomenon, that is real-time PCR and ELISA. We could find that when these knockdown cells are treated with anti-TIP1 antibody, there is no upregulation in the mRNA expression of MITKIND. That means when beta-catenin is knocked down, there is no upregulation of MITKIND upon anti-TIP1 antibody in both at the mRNA level or at the protein level. So we could conclude that yes, MITKIND induction following TIP1 blockade is mediated by beta-catenin. So I'd like to conclude this work by saying that CRISPR knockout of TIP1 results in significant reduction in viable cancer cell population. Anti-TIP1 antibody reduces cell proliferation. MITKIND is upregulated following TIP1 blockade, and beta-catenin interacts with TIP1, and blocking TIP1 results in activation of beta-catenin. And knockdown of beta-catenin prevents MITKIND expression upon TIP1 blockade. Our future work plan includes targeting TIP1 along with MITKIND so that a better therapeutic efficacy can be obtained for NSCLC. So with this, I would like to conclude my talk. Thank you all for having me. I'm happy to answer the
Thank you, Dr. for the nice presentation. If anybody has any question, then you may ask. One question.
Hello,
Yes.
it's not a query actually. We have given a very nice presentation in which you are trying to find the causes of drug becoming resistant. And I'm Pakistan, hope you remember me. We have met in the 7th folk meeting. Meenakshi, do you remember
Yes.
me?
Pakija?
Yes, yes.
Hi, Pakija. Hope you're doing good.
Hi. I am an assistant professor now here in this college. Nice to see
Oh,
you.
congratulations. Thank you.
Nice. Okay. Hope you are doing good.
Yeah, thank you. I'm doing good and glad to know that you have joined there.
Thank you. Okay, then we will talk later.
Yeah, sure.
If there is any query.
So, thank you, Pakiza, and thank you, Dr. Meenakshi, for your nice presentation. And we are going to start, invite another presenter. And I'd like to call Sahib to introduce our next speaker, Kongur Satya, for this session.
Our next speaker is Dr. Ankur Saikia sir, joining us from Copenhagen, Denmark. He is a trained biochemist with in-depth knowledge of MHC class 1 protein refolding, purification, and in vitro and ex vivo assay development for drug discovery platforms. He did his PhD from Jacobs University, Bremen, Germany, and is presently working as postdoctoral fellow at Technical University, Denmark. He is an independent and detail-oriented scientist with 12 years of industrial and 7 years of academic experience in protein biochemistry, bioassay development, and lab automation for HTS platforms. He has been awarded the award for 2010 by Julian Biosystems India for the year 2013, by Advanced Therapeutics Limited, and for the year 2016 by Syngene Limited. In 2017, he has been Explorer Award by Nehru Chapman Therapeutics. Now I request Dr. Ankur Saikia
Blink
to
one of the blink.
present.
অলপ দেৰি ব্লিংক কৰিব টানকে নিজে নিজে ষ্টপ কৰিব পাৰিম ঠিক আছে সব ঠিক আছে চলিছে ন
Hello, thank you for your nice introduction. So let me share my screen first. Okay. Okay, am I audible?
You are totally audible.
Yeah, okay. Thank you. So first of all, I would like to thank the organizer of this international conference for inviting me and allowing me to share some of my research. Here today, I'm not going to talk too much detail about the experimental stuff, but it's more or less like an overview of the PhD research that I have done here, so that you can get an idea of what I'm doing here and how we are developing technologies for the treatment of cancer using immunotherapy methods. So as the statistics says that cancer is one of the most prevalent disease in the world, with 1 out of every 3 individual getting diagnosed with the cancer, and then cancer. And out of these people, almost 50% is the survival rate, and 50% could not make it. And the people with the cancer has lots of mutations in their body, which can happen in any part of the region, and so this cancer is a very random process. It is a accumulation of genes at many different regions of the body, which can happen in any region, and so we don't really cannot say cancer is one disease, but it is the accumulation of many different diseases. This happens because normally under the normal condition, our immune system can detect like 99.999% of these mutant cells, but then a very small fraction of it, like 0.001%, escapes this immune system, and this can result in the cancer. So cancer can be treated in many different ways, like in the traditional cancer therapy, we use the radiation or the drugs that can be linked with a chemotherapeutic agent. And so in this scenario, we normally target the fast-growing cells, and as a result, it can target the healthy cells as well as the cancerous cells. So it is not very specific, and due to this, the traditional cancer therapies have lots of side effects. Whereas the new emerging field of cancer treatment, as we call it, as the cancer immunotherapeutics, they basically are like here, the immunotherapeutics unleashes the patient's own immune system. So it basically try to tweak the immune system, try to boost the immune system in such a way that it can find the cancerous cells very, very specifically and at the target. And so as a result, there is very, very fewer side effects. And also this way, we can develop a memory for the future possibilities, future infections. So if we look at our immune system, it is broadly divided into two parts, the innate immunity and the adaptive immunity. So innate immunity, let me take my, yeah. So these are the cells that comprises of the innate immune system. Usually this innate immune system is not very specific, but they could not differentiate between different pathogens, but they can just detect that there is a non-normal things happening in the body. There is a infection by a pathogen, virus, or there is a cancer happening, and then they directly act upon, and then they start killing the infected cells. Now the adaptive immunity is quite specific for specific in a sense that this immune cells has the capability to detect the specific receptors or specific antigen on the surface of the infected cells. And this is a process called the antigen presentation, which is mediated by the cells like macrophages and the dendritic cells. So if the innate immune system cannot prevent the disease from occurring, then the adaptive immunity is kicked in. So usually the most important fighter of the adaptive immune system is called the T cells. The T cells can actually find the cancer cell, like say, for example, in this case, if they see the cell with a cancer, they can bind to the cancer cell using a specific receptors, and then they can start killing, they start secreting some of the enzymes, some of the stimulants like the cytokines, which results in the killing of these cancerous cells. This is illustration where you can see that this T cells, when it binds to the cancer cells, it can start killing the cell almost immediately. Now the question here is how do these T cells differentiate between the normal cells and the infected cells or the cancerous cells? Well, this is actually helped by a very specialized immune receptor that we call as major histocompatibility complex class I molecules. In short, I will be using the term MHC1 proteins, or we can also call them as class 1 molecules. And these molecules are actually it's a trimeric molecule who is composed of a heavy chain, as shown in the red color here, and a light chain, also known as a beta-2M, and this heavy chain forms a peptide binding groove here, where these antigens from the viruses, bacteria, or the cancer cells can bind to it. For simplicity, I'll be using these cartoon models throughout my presentation. So if we look a little bit more into the details, how this MHC molecule facilitates the detection. So for example, if we consider, say, for example, we have a virus that infects the cell. So when the virus is inside the cell, because of regular cellular mechanisms, some of the viral proteins will be degraded into small peptide fragments. These are, you can also call them as the peptide antigens. So some of these fragments will then bind to the this MHC1 molecules. And once they are bound to these peptide antigens, this molecule can now travel to the cell surface, where they can present these viral antigens to the cytotoxic T cells. And when this cytotoxic T cell detects that there is a foreign antigen presented, so they can start killing these infected cells. Same is the case with the tumor cells. So now this is we know. So now we have seen that how the T cell detects the cancerous cells. But what happens in case of tumor? How the tumor escapes the immune system? Normally here on the left side, if you see here, normally when there is some problem, there is a cancer developing under the normal process, the cells will detect these receptors are expressed, the MHC molecules then present these peptide antigens to the T cells, and as a result, they will start killing and eliminate the tumor. But in case of cancer, in case of a tumor developing, what happens is that these receptors, this is one of the way how they escape the tumor, escape the immune system. Like they downregulates these MHC molecules, and as a result, these antigens cannot be presented to the cell surface, and so the cell, so that T cells cannot detect them. And also there are many other regulatory mechanisms that kick in. So these tumor cells start recruiting some of the inhibitory cells or some of the regulatory cells, which then start secreting certain chemicals that inhibits the activation of these T cells. So in the immunotherapy perspective, what we try to do is we try to reverse back this inhibition to the activation process. So basically, we try to either inhibit some of the regulatory molecules, or we try to target certain markers on the tumor cells using the T cells, so that we can kill the cancer. So the tumor in very briefly, so the immunotherapeutics can be divided into these categories. Like we can the adaptive cellular therapy that I will be talking a bit in my later slide. And then there are these oncolytic viral therapies, where these viruses are designed in such a way that they can detect and kill the tumor cells. And then we can also use the peptide vaccines, cancer vaccines, which can boost the immune system. And there are specific antibodies, monoclonal antibodies, bispecific antibodies that can also target the cancer cells very specifically. And then there are the different immune modulators which can also boost the immune system. And also there are checkpoint inhibitors that can bind to the inhibitory molecules on the tumor cells, and which results in the clearance of the T cell so that they can come in and they can kill the tumor cells. So this is one of the example where we use the adaptive cell therapy for the cancer treatment. This can be done if the tumor cells are isolated from the patient. There are some of the T cells that are still inside the tumors, although they may not be functioning with the full potential because of the suppressive environment. These T cells inside the tumor are called as tumor infiltrating lymphocytes. In short, we call them as TILs. These TILs, when they're isolated from the tumor, and then we expand these TILs, and then we inject it back to the patient, so that now they are in large number and they are more activated, and as a result, they can now go back and kill the tumor cells. There is another way of adaptive cell therapy where we isolate the T cell from the blood of the patient, and then we genetically engineer the T cell with the T cell receptors, or another method called the chimeric antigenic receptors. So basically what these receptors does is that they target certain antigens that are expressed on the surface of the tumor cells, and these modified T cells are then expanded and then again fused into the patient. As a result, they can now function optimally, and then they can kill the cancer cells. Now, what is important here is that these antigens, like the antigens that are presented by the MHC molecules. So as we have seen already, that when the T cell detects this antigen, they can kill the tumor cells, right? So the thing is that the T cell has a specific receptor here that we call as the T cell receptors. And these T cell receptors are very, very specific for a particular MHC and antigen combination. So in one T cell, there will be only one kind of TCR expressed, and so one TCR can only detect one particular antigen. Now, in our humans, there are almost more than 10,000 known MHC1 variants. We call them as the MHC1 molecules. And these variants, so out of these 10,000 variants, one individual can have up to 6 different kind of MHC1 variants. We call them as the MHC1 allotypes. And these allotypes one allotype can bind thousands of different peptide antigens that arises from the tumor. So you can imagine that we have many different thousands of MHC molecule presenting thousands of antigens on the cell surface of the tumor cells. So to detect these different antigens, one individual have almost more than 10 billion million of T cell receptors, T cells with all these receptors, so that we can optimally detect the antigens on the cell surface, so that we do not miss out any of these antigens. So the important thing is that this is what happens in the cell. So if we want to reproduce the same model, like say, for example, if we want to monitor that this MHC with the antigen is binding to the T cells, how we do in the laboratory conditions. So this is done using the recombinant modified proteins versions of this MHC protein. What we do is that these MHC proteins are linked, produced in the laboratory, and they're linked with a label or a fluorophore, so that when they bind to the T cell, we can track the fluorophore, and we can detect that they're binding to the particular T cell receptors. So this is again another graphical representation how these MHC molecules are made. So this MHC, we say, for example, we have a monomer, and it's linked to a fluorophore. And then we want to detect the TCR binding. The problem here is that when we use one single monomer, the binding, because the affinity of the monomer to the T cell is very, very low, so the half-life of binding is around 10 seconds. So probably if we want to track this using an instrument like a flow cytometry, this is not going to work, because they will wash off during the processing. So what researchers have developed is that they have designed the multimeric versions of this monomer. We call them as MHC multimers. For example, here, this is a tetramer which has 4 MHC1 molecule that is linked to a streptavidin core, and this streptavidin core is further linked to a fluorophore. And because now it has more MHC molecules that can bind to the surface of the T cell, the half-life of binding is increased to almost like around 10 minutes. And this is good enough time for us to detect. This is one example how we use the MHC multimers to monitor the T cells in the patient's blood. For example, here in this experiment, we have used a two-dimensional flow cytometry experiment, and here the cells where the PBMCs, or the white blood cells from the patient blood, were isolated, and the patient, this particular patient is has this particular allotype of MHC1 molecule that we call as A2. This is A2 is one of the most abundant MHC1 allotype that are present in almost 30% of the world population. And this A2 allotype, we made the tetramer using this A2 allotype for 2 different kind of virus antigen. This is one of the most common virus called human cytomegalovirus, and then we also made the tetramer using the antigen from the HIV. As you can see here on the x-axis, we have the signal that we receive from the tetramer, specific for the particular antigen, and on the y-axis we get the signal from the antibody against CD8, which is expressed on the surface of the CD8 positive these T cells, which are also called the cytotoxic T cells. So when we see such graphs, we can say that whatever so you can say that all these small dots here, so each of the dots represents a single cell. So whatever we see in this area, in this zone, so these cells are positive for the CD8, whereas whatever we see in this zone, these are positive for this particular tetramer. As you can see that out of the many CD8 T cell population, some of them can bind to this specific tetramer that arises from the ICMB. Whereas this particular patient, PBMCs do not, T cells do not bind to the HIV antigen. This means that this particular patient was never exposed to the HIV infection. So this is the way we monitor the T cells in the patient blood. But the monomers, the multimers can be of different types. Like they can be dimers, tetramers, pentamers, dexamers, depending on the affinity that we require and depending on the applications that we are working on. And basically, these MHC class 1 molecules are really plays really very important role in the immunotherapy. And my entire research during my PhD was based on this MHC1 molecule. So I was dealing with how this MHC molecule can be prepared, generated more efficiently, and how they bind the different antigens. What are the thermodynamic parameters that regulates the binding of MHC molecules. So this MHC class 1 multimers can be used for immune monitoring, as we have discussed previously. And then they can also be used to detect the discover the neoepitopes, like this new epitopes that binds to these tumor cells. And then we can also use these MHC1 molecules for T cell isolation and also activation of the T cells, where we take out the if you remember that in the adaptive T cell therapy, we take out the T cells and we want to activate them. So we can also activate these T cells using this specific antigen and specific multimers. So when we add these multimers to the T cells, they bind to the T cells, and then they can activate them. Now, so this is as a part of my PhD research, I was working with the production of MHC1 multimers. So how we produce the MHC1 multimers classically. In the classical process, what we do is we produce the individual chains of the MHC1 molecule in the E. coli bacteria in the form of inclusion bodies. And then, so we mix this heavy chain, light chain, and the antigen together in one pot, and then allow them to refold for a week, depending on the allotypes that we are refolding. And then after the refolding is done, we do the biotinylation, which is necessary for the binding to the streptavidin. And then once the biotinylation is completed, then we purify the proteins using different chromatographic techniques like the size-exclusion chromatography. And then we do a quality check whether the protein is, the integrity is maintained. And then we tetramerize the protein but the main problem here is that whenever we are we want a particular tetramer with a particular peptide, so we have to start the process again and again, over and again, because the MHC1 molecules are very unstable in the absence of an antigen bound to it. So we always need a particular antigen that is bound to this. So we have to do the entire refolding process for all the different antigens that we need. This is a problem when we have like thousands of different antigens that we want to make the tetramer. In that case, we have to do the same process thousands of times, which will is a big challenge and which will probably never work for the high throughput format. So what we did in our lab during my PhD is that we introduced a particular mutation in the region of the peptide, this antigen binding groove for the MHC1 molecule. What we did is we introduced this disulfide linkase. As a result, the molecule, these MHC class 1 molecules were stabilized. They are now more stable, and they can also be now folded in the empty conformation without the presence of an antigen. So now, since we can have these empty molecules, empty MHC molecules that are stable and then that are not bound to a peptide, these empty molecules can be used to make the tetramer. And now, since we have an empty loadable tetramer, so whenever we need a particular tetramer with a particular antigen, we can just use this base loadable tetramer and add the antigen of interest, and we can immediately get our tetramer of interest within few minutes of time. So this can significantly reduce the time that is required to generate the tetramers. Now, as we know that there are a lot of T-cell receptors, and then these T-cell receptors are the TCRs are detecting the large pool of MHC1 peptide that are presented on the surface of T cells. So we want to increase the throughput of our detection in the laboratory conditions. In a way that this is one of the method that I have developed in our research. So we can now make more and more tetraploid within tetramers within a short span of time, and then we can use them for the analysis of the T cells using a flow cytometric platform, flow cytometer. Here again, still we have limitation using the tetramer because the tetramers at one time we can use maybe up to 1 to 10 different T cell receptors we can detect depending on the flow cytometer that we are using, the laser we are using. But then what if we want to detect thousands of T cell receptors in the same pot? That can actually reduce the time that we invest for our studies, and also that can also reduce the amount of reagents that we use for the detection. So that's why in my current lab, we came up with this technology that we call as the barcoded MHC1 multimers. This is one of the most efficient and most high throughput technology so far available in this area. So here what we do is instead of now linking the MHC molecule as a tetramer to a tetravidin, we link the MHC1 proteins to a dextran backbone. So here are there are lots. We can add as many as we need depending on our requirement, and each of these tetramers are assigned a particular barcode. We call it so these are like some short DNA sequences that we can later do the sequencing analysis and see that which barcode corresponds to which MHC1 peptide combination. Now with this method, instead of like 10 different TCRs, we can now detect up to thousands of, like say many thousands of T cell in the patient's blood at the same time. So this was one of the significant discovery in the area. So which was critical for the high throughput kind of studies. And then in my current lab, we also have developed certain we are developing certain technologies which again uses these MHC1 molecules, and we call them as the impact scaffold. Here what we do is we try to mimic the antigen presenting cell, where we have the T cells. We say imagine we have like a cancer cell that has MSC1 molecule presenting the antigen. But also we need lots of different co-stimulatory molecule signals that is required so that the T cell can recognize the cancer cell and get activated. So here we have developed a scaffold that has the MSC molecule along with the stimulatory signal. And when we pulse them into the T cells can get activated very efficiently, and then we can use these activated T cells again for the immunotherapeutic purposes. So these were the main technologies that we have developed in our lab. So I was involved during my PhD and now in the postdoc. And each one of these technologies actually have made into a different companies. And then so these technologies are now used by 3 different companies for the commercialization effort. And so with this, I would like to just summarize that this immunotherapy, now the immunotherapy is a very emerging field of cancer treatment that can be very efficient with lesser side effect. And then it is very important that we can identify the immunogenic antigens that can be used for the cancer immunotherapy. Using so this we do using the empty loadable MSC1 molecule, because using these molecules we can now very efficiently generate the multimers in a very short span of time. And this empty multimers or the tetramers, we can also use them to fish out the antigens from the tumor samples. Like, for example, when we swap out the tumor samples, so there in the tumor suspensions, there will be like lots of antigens that are floating around. So if we use these MSC tetramers, we can just they can just bind to the tumor antigens, and then we can take them out of the tumor cells, and then we can identify them using many different technologies like the mass spectrum tool. And then nonetheless, so we can use this MSC multimers for the evaluation of T cell and then activation so that they can be used for the immunotherapy activation. So with this, I would like to conclude my talk. So if anyone has any questions, feel free to ask.
Cancer therapy. Specifically open for discussion. If you have any questions, then you may ask.
Can you repeat the question? Actually, I could not.
The session is open for discussion. If somebody has any question, then he or she may ask you. Am I audible, Dr. Surya?
Yeah,
Am I audible?
I am not able to hear clearly. The voice is not so clear to me.
Am I audible now? Am I audible?
I am sorry. So.
Okay.
So is there a question or?
No questions are there.
Okay.
So thank you, Dr. Satya. Now I'd like to hand over the microphone to Sahil to introduce our next speaker for the session.
Yeah, thank you very much.
Thank you, sir. Our next speaker is Dr. Ramtu Saikia, sir, who is joining us from Berlin. He is an IT leader with 15 plus years of experience in enterprise customer engagements during business technology strategy. His core expertise lies in infrastructure and application migration, modernization, and cloud adoption strategies. He has worked as Senior System Administrator at TCS in Delhi and California, as Lead Consultant at Computer Sciences Corporation, as Infrastructure Lead at Bombardier Transportation TCS Berlin, as Senior Cloud Engineer at Gamigo Group AG Berlin, as Director Technology Consulting at Devo Team GmbH Munich, and presently as Partner Sales Engineer at Google Cloud, Google Germany, Berlin. He has several certifications to his name, including Google Cloud Certified Professional Developer, Red Hat Certified Engineer, Certified Linux Administrator, amongst innumerable others. Over to you, Dr. Rantu Saikia, sir. We are eager to listen to your talk.
আপোনালোকে মোক শুনিছে?
You are audible now.
Okay, just let me share my screen. Okay. Can you please confirm if you can see my screen? Okay. পোনপ্ৰথমে মই ডক্টৰ ৰঞ্জনা গগৈ বাইদেউ আৰু ডক্টৰ দিলীপ কুমাৰ ডেকাক ধন্যবাদ জ্ঞাপন কৰিছোঁ। তেখেতসকলে মোক আজি আপোনালোকৰ এই অনুষ্ঠানটোত ইনভাইট কৰাৰ কাৰণে, আৰু মই পাৰ্চনেলি আই এম pretty happy to give you a lecture on the development that is being happening with computer science, one of the branch in computer science, that is artificial intelligence. So আপোনালোকে এতিয়ালৈকে দেখিলে chemistry fieldত, biology fieldত কি হৈ আছে. So we got to learn a lot from Bhaskar, who is pioneering in the semiconductor industry, and from Meenakshi and Ankur. They are kind of doing a great work towards finding some cure to cancer treatment, right? Which is, of course, a very big disease in our humanity. So now let's try to switch gear towards computer science, and let's try to see what is happening in artificial intelligence. So this is considered to be the next big breakthrough in humanity. Okay, so just a small introduction. So there was quite a good introduction of me, but just to let you know, I'm Rantu Saikia, originally coming from Dibrugarh, and currently I'm living in Germany, in Berlin, for almost like 9 years now. And at the moment, I'm working for Google as a sales engineer. So basically, I'm responsible for most of the technical aspects when it comes to our customers within the cloud division. So I have been working in the industry for more than 15 years, predominantly with Unix and Linux systems, be it development or administration, or hosting some big ERP solutions on top of it. And lately, my focus area is data analytics, AI, and ML. So you can see there's a QR code. So I'm quite active in LinkedIn. So in case if you like to connect with me, please scan it. And then, of course, we can talk later. So today, this is the summary that we are going to talk today. So first, we will see what is artificial intelligence. And then we will have a look at different AI spectrum. And then one of the key kind of artificial intelligence technology that is being gaining a lot of traction now with that is generative AI. So we will try to see what it is and how it could basically impact our humanity. And last, we will have a look on some of the great opportunities that are lying ahead when it comes to generative AI. So with that, let's get started. So this is a slide that I have put just to kind of give you an impression what artificial intelligence is going to be in the future and in next years to come. So historically, if we see, so these are some of the technology out of many inventions which has led to where we are today. So steam power, electricity, information technology. So these are the innovations which has changed the way we work, the way we live, and the way we communicate with each other. So today, artificial intelligence is the next big thing that is going to change the way we live, it will change the way we talk, or even we work in our different industry segments. Even it will change the way most of scholars like you do research. So I will come back to that in generative AI. So first, let's try to see the definition. What is artificial intelligence is all about? So this is one of the key branches within computer science that basically deals with creation of intelligent bots or intelligent agents or machines, we can say. And these machines are trained on a large set of data. And from those data, these machines can learn and it can reason and it can act autonomously. So the key question here is how exactly those machines can do, even if we feed in the data, how exactly they can learn from that. So there are certain algorithms or certain architecture that leads these machines to learn from those data. And we will have a look in a while. So before going to that, the machine learning topic, let's try to understand the different spectrum of AI. So we have two spectrum of AI. That is, one is predictive AI and one other is generative AI. So predictive AI is when we feed in the data to a machine learning model, and then it can predict the future outcome. For example, let's say I have fed in the weather forecast for last 10, 20 years to a machine learning model. And then based on the data, the model can learn and it can predict what will be the weather for next 10 days or maybe next 1 month or next day even. And another example could be, let's say we have a college or a school, and depending on the students' behavior, the pattern with which they're learning, the way they do their homework, the topics that we are covering within the class, then what is the likelihood that how many people will get what kind of grades? So these are the outcomes that the machine learning model or the agent can predict. And so this is predictive AI. So generative AI is when the terminology or the way this generative AI works is, or the trained is same, like we have a vast set of data that we train the models, but instead of predicting the future or predicting the outcome in the future, the generative AI create the new content. For example, you can ask the generative AI to write an email for you. You just put a context, let's say, okay, write an email regarding a particular topic, then it can write the email for you. So let's go deep, let's go a little bit more deeper into these two AI spectrums. But before going into that, one of the fundamental element of artificial intelligence is machine learning. So what is machine learning? So machine learning allows a system to learn from a big set of data. So in this example, you can see how it is working, how machine learning basically works. So first, we have fed the machine learning model certain pictures here. In this example, you can see those pictures, cats and dogs. So now, first we feed the data by labeling them, like we know the answer. The machine learning model, it knows the answer. Okay, this picture looks like cat, or this picture looks like a dog. So once we train the model, and when it is ready to serve, then we just pass on any picture, maybe a cat or maybe a dog. Then based on its historical learning, it can recognize a picture and it says, okay, yes, this is a cat. So this concept applies for all kind of artificial intelligence. So basically the base of everything is machine learning, and that is data. So unless we have unless we have enough data to train the models, we will not be able to succeed in creating some sort of, let's say, a artificial intelligence agent or program. So here you can see some of the examples. So examples of like machine learning models that we have. Linear regression model. This is a model which is mostly used for the example that I said. It takes certain input variables and it gives an output. For example, the passing or the grade score prediction is a linear regression model. Then we have logistic regression model, which is basically where we say that what is the chances the customer will buy a certain product. So this is another example of logistic regression model. And the classification model. So this example that you see, cats and dogs. So the classification model does this classification whether one particular object belongs to a particular type. Then we have the artificial neural network, ANN model. So this is one of the most advanced model that we have. And this is basically used in generative AI, which I will talk later. So when you talk about different spectrum, this is computer vision. This is another great achievements or inventions that we have today. So computer vision is the ability of a machine to see and understand the world around it. So as a human, of course, we have our eyes and we can see and we can take a decision like, okay, whether to go right or to go left. But what about the machine? So here we train the machine based on the data that it sees around itself. For example, one of the very big use case here is self-driving cars. So self-driving cars are dependent on vision, computer vision, meaning it is able to see what's on the road. It can detect a human and it can detect the traffic lights, traffic signals, or road signs as well, and it acts accordingly. Of course, self-driving is not only dependent on computer vision, but it also depends on a lot of other factors as well. For example, it has inbuilt radar, it has of course cameras, and it also has other technologies which basically helps it to make a decision whether to take left, right, or of course when to stop. And another few examples of computer vision are, let's say, facial recognition. So if you use Google Photos, so if you tag somebody, a person within the photos, and if you search with that tag or with that name, then you will get all the photos related to the person. So this is basically done through facial recognition. It's also, of course, used in security as well. And another interesting use case is medical imaging, where the disease could be detected in a very early stage through imaging, and then of course then we can have a better cure for that. So next I would like to talk about another spectrum of AI, that is natural language processing. So here the NLP is the ability of a machine to understand, respond to human language. So this is basically widely used across speech recognition, and then translation. So maybe if you use Google Translate to translate certain languages, that behind the scene, this is NLP is the model that is running. And of course, in this, you can see this the example here, I'm sure some of you might be using Gmail as your email. How Google is basically detecting the legitimacy of an email, and it is detecting as a spam or it is a legit. So the machine learning model that is running behind it is NLP, natural language processing. So then comes the technology that is kind of gaining a lot of traction now, which is Gen AI, generative AI. So as I said earlier, the significant difference between a standard or predictive AI and the generative AI is its ability to generate new content, not only text, but images, audio, and video. So meaning you just need to type in certain keywords. Let's say, okay, give me a recipe for cooking chicken, for example. So it will give you the recipe, which is kind of widely used. Or even you can say, okay, give me a recipe of cooking Assamese chicken, because Assamese chicken curry is different than the other chicken that you can find in maybe South India or North India, right? So it takes the input and it gives you the output as a new content. So in this example, you can see you can also write an email, like you can formalize the email. So you just put in the key topics, let's say the subject line of the email, and then you just click on formalize. Then it will write the entire context, and then you can just check, proofread it, and then you can send it. So how exactly the generative AI is able to do that? So there are kind of two fundamental things that we should know while talking about generative AI. So large language models and transformers. So what is LLM? So LLM is basically this is a large language model. It's a type of artificial intelligence that is trained on a massive data sets of text and code. So this is the key thing to remember. It's a text and code, and this allows the models to learn the statistical relationship between those text and the code, and then it can kind of create the new content based on those understanding from those huge amount of text and code. So some of the industry-leading example of these large language models are Google has it's called Lambda, so language model for dialogue application, and OpenAI GPT-3. So you might have heard a lot about ChatGPT, right? So behind the scene, ChatGPT runs GPT-3 engine that is generative pre-trained transformer 3. So these are language models that is responsible for creating those new content within generative AI. Another key thing is the transformers. So transformer are, you can say, the subset of LLM. So LLMs use transformers to kind of parse those text and code. So transformers are basically neural network based algorithms, and the key thing is it understands the sequences of the words, phrases, and learns the context. So because of the sequence, and it can learn the context, because so what I mean by context is basically when you ask the model certain questions, it will remember the context. So let's say if my context is, for example, context could be my subject could be, let's say, I'm trying to learn more on a particular topic, let's say animals. So whatever questions you ask the model, it will try to remember the context that is animal. So it will try to give you the output based on that context. So my next example within the generative AI is some images. So I will show you some images which are basically not drawn by any artist, but those are kind of created by generative AI technology. So maybe I will switch to Assamese, because that will give more kind of feel to explain what it is. So আমি সৰুৰ পৰা আমি যেতিয়া ডাঙৰ সৰুৰ পৰা আমি যেতিয়া সাধু কথা শুনো আমাৰ মা দেউতা বা আমাৰ আইতা ককায়ে কয় বুঢ়া ডাঙৰীয়া বাঘ পিখাচ সো এইবিলাক আমাৰ সাধু কথাৰ ক্যাৰেক্টাৰ ৰাইট সো আমি কিন্তু আমি কেতিয়াও এইকিটা ক্যাৰেক্টাৰ আমি মেবি আমি মনত ভিজুৱেলাইজ কৰিব পাৰো কিন্তু আমি চকুৱে কেতিয়াও দেখা নাই আৰু কোনোবাই যদি দেখিছে মিছা কথা কৈছে So, Kintu, it is a generative AI help. So, using generative AI, one of Assam's profound filmmakers, Sinmoy Burma, so tekhete ki korile, tekhete Midjourney boli eta generative AI tool ase platform, midjourney.com. So, tat goi tekhetor imagination text input dile a kita character and the generative AI, the image Midjourney has given the pictures. These are the pictures that you can that it has kind of responded with. So you can think of like how powerful these tools are. So you could do really enhance and amplify your creativity skills as well. So this is just one small example with for images, but think of what you could do in maybe in your research or in your let's say you want to write a book, so you need some ideas. So this tool can give you a lot of ideas about any subject that you would like to write a book on or do some research on. So now, of course, this tool is really powerful and this is going to change the way we work in every field. So, but of course, there is a drawback to it as well. We all know everything has its good and bad side, right? So there might be people who try to exploit this feature and maybe try to use it for their own maybe some bad things, right? So that's why the one thing that we need to understand at the very beginning of AI revolution is that we have to be responsible. So any step that we take today should be for betterment of our humanity or betterment of our profession or any field that we can think of. So far, when it comes to generative AI, so these are the opportunities that lies ahead of us. So these are some of the personas like analyst, customer service, creative writer, or creative thinkers. So for all these people, generative AI can make a huge difference in the way they work today. And I'm sure going forward, as we kind of, as the generative AI evolves, you will kind of experience a lot of differences, the way you use your smartphone, the way you kind of, the way you maybe browse the internet, the way you do an online shopping, or the way you use any machine, any computer device. So with that, that's the kind of end of my small presentation. So I hope you like it, and I would be happy to take any questions if you have, or feel free to connect with me later as well if you have any questions around this topic, or if you'd like to know more on this. Thank you so much.
Thank you. This is okay. Nice presentation. Now the session is open for discussion. If anybody has any questions, then you may ask session again. So I think no question. Right? Anybody wants to ask? Danke. Rangu. Very nice. Danke, great.
ধন্যবাদ দিলীপ স্যার।
Thank you. Next will be announced by our chairperson, sir. So thank you, Satya. So সুন্দৰ presentation দিলে, শুনা পাইছে? Okay.
মই
শুনিছে?
দেখা পোৱা নাই।
Anyway, I think no questions are there from spectators. So I'd like to hand over the microphone to Sahil to introduce our next speaker, Rana Hawaldar. Sorry, Rana Hawaldar.
Thank you, sir. We now have an alumni of our own department, Department of Chemistry of Borno College, and presently a senior process scientist at Applied Material Incorporation, Santa Clara, California, USA. Dr. Rana Hawaldar, sir, has achieved CSIR JRF in 2005, GATE in 2006, CSIR SRF in 2008, and DST Travel Award to attend ACS conference held in Anaheim, California in 2011. He also has numerous publications in reputed journals. He has attended several conferences in India and around the world and has multiple patents such as selective silicon dioxide deposition using phosphonic acid self-assembled monolayers as nucleation inhibitor and many others to his name. We are delighted to have him here, sir. He has 10 plus years of industrial experience in nanotechnology, clean room process development, organometallic synthesis, and thin film deposition techniques. With this brief introduction, I request sir to kindly deliver his talk now. Over to you, sir.
Thank you very much, everyone. I think I'm audible. Can you hear me?
Yes, sir, you are audible.
Yeah, you're clearly audible.
Okay, thank you. Just give me a second. I just try to, share my screen here. Give me a second, please. Bear with me. Okay. Can you see my screen by any chance? Just let me know if you do. Can you see my screen? Can you confirm, please?
No.
Okay.
It is not.
Screen con visible one.
Oh, sure. Just a second. Share your screen. Whiteboard. Why? Advanced files. Oh, something wrong.
It's not Teams, Rana. It's Zoom.
I know it's Zoom.
Share screen, that's at the bottom, right? And third one, fourth one from right.
Which one, Bhaskar? Can you just guide me?
It's the fourth icon from right side bottom, or kind of at the center of the screen, right? The bottom.
This one?
Yeah, share screen.
No, it's getting some error, actually. That's the problem. Actually, this is my personal laptop. Okay, Bhaskar, question. Did you share it from the company? Did you use your company laptop?
Yep.
Okay,
You can use that.
just sorry, guys. Sorry, team. Actually, give me a few minutes, okay? I'll transfer it to my office laptop. I think it's some issue with the laptop here. Just a second. I'll be back.
So while you are waiting for this for Rana, right? So I have a quick question for the organizers. So I mean, is it a conference that you guys have like every year, or you guys are kind of starting it up now?
Sir, am I audible to you, sir?
Yep.
Yeah, actually, we have started this. This is the first international conference, and we are beginning to organize such international conferences at our college. So this has been organized for the first time here at Goga
Oh.
College.
Okay, got it, got it. Thank you.
Thank you.
And what is the audience? Like, is it your master students or PhD level student, or what is it?
Mostly the audience members are undergraduates, and some of the research scholars have joined us from different institutes to participate in the conference. So we can say that it
Okay.
is a mixed audience, having mostly faculty members of our college, research scholars, and many undergrads.
Okay, got it, got it. So I mean, for the especially for the undergrads, right? It has to be like more generic, I guess. That would be more helpful, I guess.
Yeah, right.
Okay, so I just want to comment one thing. So, so for the undergraduate students, do you have any questions for us?
Pardon, I couldn't follow your question. Did you ask about
So
fellowship?
maybe I can also talk in Ohmia, like undergraduate.
We all can talk in Ohmia, by the way, right? সকলো অসমৰ হয়, like somewhere from Dibrugarh,
Mainly undergraduate students, মানে students যোনবোৰ আছে তেখেতসকলৰ কাৰণে মই সুধিছো মানে কিবা question আছে নেকি আমাৰ মানে career related or anything কিবা জানিব বিচাৰে নেকি আমাক কন্টেক্ট কৰিব পাৰিব পিছত এতিয়া নহলো পিছত হলো LinkedIn profile চালে LinkedIn থ্ৰুদি কন্টেক্ট কৰিব পাৰিব মেইল কৰিব পাৰিব দৰকাৰ হলে ফোনৰ থ্ৰুদি কন্টেক্ট কৰিব পাৰিব মানে ফিউচাৰত যদি কিবা প্ল্যান আছে যে বাহিৰত অহা বা বাহিৰত ৰিসাৰ্চ কৰা
Dr. Rana. Dr. Rana.
He is Ankush Shekhar.
Hi, Dr. Hokia. আচ্ছা এদিন এটা সেশন স্পেশালি তোমাৰ কাৰণে অ্যারেঞ্জ কৰিম career oriented এটা সেশন
yeah, get ready.
One full session will be dedicated to Hokia. Okay. Eta full session rakhim. And Dr. Bhaskar, am I audible to you? Yes. এদিন ভারে দুদু এদিন তোমার কারণে এটা সেশন রাখিম দেই স্পেশাল সেশন তোমার কনভেনিয়েন্টলি
মেবি আমি সবে একেলগতে আহিব পাৰো দৰকাৰ হলে মানে
by the way, right? I mean, again, this is a chit chat. Ankur আর রন্তু আমি স্কুলত একেলগে পঢ়া আমি গোটেইটা
ওকে অংকুর রন্তু সব সৈনিক স্কুল গোৱালপাৰা yeah all boys
yeah. So Meenakshi again we একেলগে আছিলো ইয়াত California, I mean California.
Meenakshi is there actually. Meenakshi আছে গতিকে আমি এটা সেশন ৰাখিম স্পেশালি ফৰ কেৰিয়াৰ কাউন্সেলিং আমাৰ যিখিনি আন্ডাৰগ্ৰেজুৱেট ষ্টুডেন্টস থাকিব বেসিক্যালি সায়েন্স ষ্টুডেন্ট এইখিনিৰ কাৰণে এটা কেৰিয়াৰ ওৰিয়েন্টেশন প্ৰগ্ৰাম ৰাখিম ফৰ স্পেশালি এটা সেশন থাকিব ফৰ সায়েন্স ষ্টুডেন্টস গতিকে মই আশা ৰাখিছো ডক্টৰ ভাস্কৰ ডক্টৰ ৰন্তু ডক্টৰ অংকুৰ ইজ দেয়াৰ and I think Dr. Meenakshi will also accompany us. Then it will be a very, very beautiful session. That will be on career counseling option.
Sure.
Okay,
Yep, yep.
that's fine. That's fine.
Runner কি হল?
Rana is not there. I think he is busy with other job. Okay. Rana is not there. We have another session, but it's another link. It is in another link. That session is also chemistry, but link is different. So,
ৰানাৰ কিবা টেকনিকেল প্ৰব্লেম হৈছে বোলে so ই মানে solve কৰি আছে
ভাস্কৰ
অ
ৰাণা আমাতকে জুনিয়ৰ আছিলে নেকি নে কি আছিলে বাৰু
ৰাণা আমাৰ লগৰে
ডাঙৰ
লগৰে হ'ব প্ৰায়
বহুত দিনৰ আগতে লগ পোৱা তাক
মুম্বাই ডেইজ ন So, question for the organizing committee, right? So, was this talk, I mean, were this talk like helpful? Do you think it is helpful for somebody like the students mainly? Or maybe it should be more generic from next time, I mean, if we get an opportunity again.
Right, sir. The sessions were particularly helpful. Some of them were more technical than the others, but on the whole, the sessions were very interesting, and we could see the work that you are doing. So.
Got it. Yeah, I think the idea should be like more about that is more beneficial for the broader group there, audience there, right? I mean, if it is the undergrad, like if we know that all those details prior, I mean, that will be helpful for us to make the presentation. I mean, from next time onward, just in case if you guys invite us or whoever, right, next time. Because I knew, I mean, what I heard was like there will be some PhD students kind of would be the mostly dominated by that kind of population, but it seems like you still have undergrads, right?
So, sir, I think it will be particularly interesting when the students would be able to directly interact with you if we have a session with, say, one of the speakers at a time, and they may be allowed to interact with you one at a time.
Sure, sounds good, yeah. So yeah, actually, I go to Assam, I mean, like I go to Sibsagar all the time whenever I'm in Assam, so maybe I can go down to the college also in person.
That will be very nice, sir. Yes.
And again, don't call me sir, just call my name. That's how we go here. Not used to being called sir.
Rana একই ছেলে।
I think Rana is here.
Can you request for the control, Bhaskar?
I don't even know how to request control here. So there is a button at the bottom, right? So to share
Right.
screen, you can see mute, start video, security, participants.
No, for
So
me, all are errors. So that's the problem I have.
Can I suggest something?
Yes, Ankur.
Hi Rana, how are you?
Good, good, Ankur.
Yeah. So maybe you can mail your presentation to Bhaskar and he can
I did try that actually. That is a okay. How about this? I did try that one, but somehow
then Bhaskar can share the presentation and you can keep talking. Yeah, so it is one way maybe.
okay. Just I did try that actually. Okay. I don't know why it's not letting me. Okay.
you can do my Gmail also, Rana, if you whatever works for you.
Okay, yeah. Share present. Okay, advanced sharing option.
Maybe you can also use Google Cloud. Rantu's Google Cloud.
Okay, Rantu, can you request access to this one? Actually, I don't know. Actually, I'm not used to this Zoom actually. We are team users, so
Maybe he is going for breakfast. He's not here, I guess.
just a second. I think I just give me a minute here. I think I'm share screen, share presentation, advanced basics one. Microsoft Share. Can you see my screen now?
No, sir.
Okay, somehow from this I'm not able to send to Bhaskar as well. That is okay. Place OneDrive personal.
So Rana, when you click on the share screen, what do you see? Like with the green button, what do you see?
Okay, let me, Ankur, okay, so can I do a video call from my cell phone and then, okay, in that case, let me do it from my cell phone, right? Issue is that I'm not able to, oh, okay, just a second.
Rana,
Sure. Big
তুমি যদি
problem.
PPT খন মোৰ তালে পঠিয়াই দিয়া, মোৰ
হয়,
WhatsApp
sir.
পঠিয়াই দিয়া, মই
হয়, sir.
ইয়াত দেখুৱাই দিম।
ঠিক আছে স্যার, মই হোয়াটসঅ্যাপত পঠিয়াই আছো ৰ'ব আপোনাক। ওকে,
তুমি মোলে WhatsApp কৰি দিয়া,
ঠিক আছে স্যার।
মই ইয়াৰ পৰা শ্বেয়াৰ কৰি দিম।
Share.
Hello.
ভাস্কৰ, মই মোৰ স্ক্ৰীনখন তোমাক দেখাই আছো, ঠিক আছে? কিন্তু সমস্যাটো হ'ল কি, তোমাৰ মই এই স্ক্ৰীনখন তোমাৰ শ্বেয়াৰ কৰিব পৰা নাই বা তোমালৈ পঠিয়াব পৰা নাই তাৰমানে। এক ছেকেণ্ড ৰ'বা।
Hello, am I audible?
Sir, you are audible. Somehow I'm not able to share, I'm not able to send this file to any email too. It seems like
Okay,
the
okay, no problem.
it
We
got
are,
corrupt, it seems like. Portraits share.
now we are going to conclude this session, and we have another session for chemistry. So I request you to join in the next session. I will
sir, that is fine. Let me
send
prepare
you
this out.
the link, Zoom link for the next session. This is especially for the chemistry. Okay.
Okay, sure, sir. Let me try to.
So
figure
I
it out.
request Rana to join the next session. We'll share our Zoom. Okay. So now I would like to I request our chairperson of the session to announce the conclusion of the. Okay.
So I really apologize for this inconvenience to everyone. Actually, I'm really sorry for this one.
So, no problem. Can you hear me? Okay, Dr. Halada, it's not a problem. Due to some technical problems, we have to stop the session, this technical session. for
Okay.
now.
Okay,
Next
I'm gonna.
session And secondly, I have
Sir, I'm going to record my actually video, whatever presentation, right? And then share it to you. Maybe let's see, let me try that one meanwhile. And then you can proceed, right? Yeah. Let's not wait for this one. Sorry for waiting.
I think this session is over, right?
I hope so. Maybe we can drop off.
Yeah.
Yeah. Okay. Thanks, guys.
Bye.
Nice meeting you.
Thank you,
Thank
everybody.
you.
Bye.
Have a good night,
Bye.
good day, whatever it is.
Have a good day and good night, everyone.
Midnight. Bye.
Bye.
Itu apa sesungguhnya?